Greetings, everyone, from my home, a little outside central London. A lot of us can't be in the museum more than a few days a week because of the COVID regulations. And we're all looking forward to being back amongst our charges as soon as possible, possibly in late April. So I'm going to share with you really some impressions that I have from London, impressions from London of a collection that is intended to be useful to people all over the world. Some of you will have been able to visit us or make inquiries of the collection, and I'm going to give you some details that you might find interesting. So next slide, please. I'm very fortunate to be surrounded by this massive collection. 250 years of accumulation of some of the most important and varied specimens of fishes in various different states of preservation. And my job is to look after them and to make sure that all of you can use the enormous resource that they represent. Next slide, please. In 1973, in fact, it was, Rajiv, that I interviewed for what was basically my dream job. As soon as the advertisement appeared, all my friends started writing to me and saying, look, you've got to go for this, because I had always been so fanatically interested in the museum and in natural history. And yeah, on the 24th of August, 1973, I got this wonderful letter saying I'd got the job. And so I started. In those days, we had long hair and flared trousers, if we were even at all up to date. And I learned, as you can see, how to carry a lot of specimens around under the watchful eye of Peter Whitehead and Owen Wheeler, who were my bosses in the marine fish section. We had a marine and freshwater subdivision in the group, although that didn't make, that wasn't hard and fast, as many groups, as you know, are. Live in both freshwater and salt. And I joined this team. So there were 11 of us, or 12 of us, and unfortunately, I think only 4 of us are still alive. But this was a great team, really pursuing the same research agenda that Gunther and Tate Regan had set up at the beginning of that century. So it was very exciting to join this team, to become involved in all their projects and to learn the tools of the trade. Gordon Howes on the left, myself, Ethel Winter-Warvis, Peter Whitehead, Margaret Clark, Jeff Palmer, Humphrey Greenwood, Keith Bannister, Rosemary Lowe McConnell, Owen Wheeler, and Jim Chambers. Great team. So here's a few figures. It's a massive collection, 500,000 fluid preserved specimens. And the various categories of material you can see here, the 4.5 kilometers of shelves for the Spirit collection give you an idea that, you know, it really is, in terms of the sheer mass of material, a very important collection. And there is no doubt that Asian and Indian fishes form a very important section of that collection, partly because of British involvement in India and because of the work of a few very industrious collectors. So when I joined, the only way that we kept the records of the collection and the details of the specimens that were in it were by handwritten accession registers, following on from the published 8-volume catalogue of Albert Gunther. And so the accession register started in 1837, and the details of each specimen were recorded in these ledgers. We had to write by hand, and during my interview, one had to produce a handwriting sample during the interview and push it across the table to see that they wanted to see that you weren't going to mess these books up. And so that's my first register entry page and it was unfortunately, a few crossings out until I got my nerve. And this was actually on the first day of my working in the museum. That's not me on my first day, but the page is open at my first register entries. Of course, this isn't very accessible, this kind of record, and inquiries as to regional or systematic categories of material in the collections were tricky, because you had to work your way through these registers and to pull out the information. There were systematic catalogues as well, also handwritten, in which we could gather the material, gather the information systematically. These are all type specimens, just one shelf within the type series. The types are segregated from the main systematic series of collections, because they are the first priority in an evacuation. In the Second World War, these were all taken and stored in underground caves in case the museum was bombed. So yes, these are our crown jewels, the reference specimens for the taxa that relate real animal bodies to names. And of course, they're the way that we ensure that our progress expands outwards, and we're not going backwards. This is how we keep a track of what we do know in order to advance our knowledge. The types are what people come to visit the museum for, and they are really the most called upon for data as well. The spirit collections are much bigger than just the types, of course, and this is a view of one of the storerooms. We have 3 floors like this, and they're kept at 13 degrees centigrade because we've got this fishes stored in alcohol. That's a method we know has worked for 250 years. We hope, using the right methodology, we can keep it going for another 250. But this storage in relatively dark conditions in the cold is ideal. That can't be done everywhere in the world, we understand, and but it has ensured that we've kept the material that we have in good condition. And of course, the collections are systematically arranged. The systematic arrangement can only be that at the time that they're shelved, and we cannot really physically move the collections around as our knowledge gives us new classification ideas. So you need expert guidance to get through our collection. It helps to have people who have seen some of the changes that have occurred over the time of the storage of the collection, so that we can point you to the material. A collection of this size is always going to be difficult in some ways, because as our ideas change, but the specimens stay on the shelves. We can link modern ideas to where the material really is. We not only have to have some understanding of the systematic coverage of the collections and the actual specimen locations, we've got to look after them. And the collection is a real long-running experiment in what works. The materials that some specimens have become compromised over time, so we know what doesn't work as well. It's necessary, looking into the far future, to make sure that we're using the right methods of preserving the fishes. And so that includes, in this case, actually having commissioning glassware for our purposes. We have special needs, unlike a lot of industry, and we need very close fitting, sealable glass containers. Glass lasts for a long time. Metals and plastics don't do so well, especially in an alcohol vapor environment. So for long-term storage, it pays to get the right materials in the first place, and we commission our containers. For larger material, larger specimens are always underrepresented in museum collections. It's easier to collect small samples than great big bodies. And we have these stainless steel tanks now, which enable us to preserve large specimens in a very stable way. And it has meant that when very large specimens become available in the UK, for instance, we're very often the only people who can preserve them for science, and sometimes for the first time. So the alcohol preserved material collections are massive, and that the longest tank there contains a large Greenland shark, which we got a few years ago. That's, I think, the only adult Greenland shark preserved, certainly in the UK, in spirit. Next slide, please. Skeletons, another very important collection, made either by boiling the specimens or putting them in hot water, and then you get a disarticulated skeleton, very useful in anatomical studies, and archaeologists find them useful as well. Articulated skeletons like this Nile perch are made using our domestic beetle colony, which eats the flesh away for us and provides an articulated skeleton. This way of looking at osteology has not been entirely superseded by modern methods, but we have CT scanning and X-rays, but looking at the bones in the round is still very important from a comparative anatomy point of view. Next slide. Before the beginning of the 18th century, when alcohol preservation started to be developed, the only practicable way to preserve animals from around the world and bring back specimens was to dry them. And we have a huge collection of stuffed and sometimes skins mounted on herbarium sheets. These dried specimens still contain a lot of very useful information, even though the internal anatomy is mostly missing. And of course, many of them are types. These are still the specimens that have to be referred to in our elucidation of biodiversity. Next slide, please. Skins, sometimes dried out on herbarium sheets, sometimes with a little padding inside, are another form. The Day collections, Francis Day's collections, are usually mounted on herbarium sheets, some of them not. And these specimens are still referred to quite regularly. They're not, in any sense, outdated collections. Next slide, please. This will be perhaps a nostalgic sight to some of you who've worked here. This is the visitor's home when you're working in the collections. We've got a few comfy chairs there and some now slightly dating binocular microscopes. And this is where the material is bought and where you work on it. And people from around the world have come to use our collections in this space, and we welcome you. I'm thinking about some of the types of work that I do and my colleagues. We have backlogs of unsorted material. Every large museum does, and important collections are still in need of sorting. And this, I see, was this is a 1960s collection, and we, as a background activity, we can use this material to train students and volunteers in the art of keying out and sorting material, and it's a collection building exercise which can continue as a background activity. This is a chart of accessions from 1837 to 2017, and annual numbers of specimens added to the fish collection. You can see some historical signals showing up: the 2 world wars, the big discovery collections, accession. But you can see that from 2,000 to 4,000 specimens a year is a sort of level that we've maintained over a century. A lot of the work in keeping this material secure is quite manual and repetitive. Our enemy, evaporation of alcohol, is something that we're constantly battling with, and although we make sure that that's kept to a minimum with this low temperature storage and good containers, we keep our eyes on the levels of alcohol, and we're always topping them up. We have a lot of manual collection moving to do, and every now and then we've had to move to a new building, and one wants to do that as little as possible. I've only had to do it once in my 48 years, and it always causes some damage to the collections, and it stops you doing the mainstream work of the of studying them. So we were out of action for a year while we moved this collection, and I hope we don't have to do it again for a long time. Big specimens, as I say, underrepresented in collections. We now, with this tank room, with our stainless steel tanks, have a good opportunity to take this material on board. So when something like this large thresher shark becomes available, we, above anybody in the UK, can say, yeah, we can take that specimen. We have to ration ourselves. We haven't that much space, but it's nice to be able to say yes when these become available. So big specimens require quite a bit of cooperation, and there's not enough people, as you'll see, in the fish team to deal with carting specimens like this around. There's some specialist equipment needed, and the curators from various groups, so mammalogists and malacologists, will all get together when a big specimen like this needs to be preserved. And we need to be in numbers because a lot of it involves quite hard work. Hefting this up a flight of stairs was quite a challenge, but if you're determined, it can be done. Yeah, a very big specimen, 30-foot basking shark. This is at the limit of what we can sort of take on board. We haven't got a whale shark in the collections. And the preparation of this needs to be very careful because the fluids, the formalin needs to get right through the tissues in order to fix the specimen. You don't want decay to continue in the interior of the body, and then good alcohol preservation as well. It requires a little skill. Next slide, please. And yeah, this beast was a challenge. We'd got mistaken information about the size of it, and so we're rather surprised to find that it was a 30-foot specimen, but we took it on board. And it has yielded interesting information already and will continue to do so. We dissected the spiral valve, which was the site of some parasites of interest to our parasitologists. So yeah, it was worth doing. This also was an extremely worthwhile specimen. Sturgeons, anadromous spawners, they come up the rivers in Britain to breed. They're very depleted in the UK. The single species that was known to occur there has virtually disappeared from British coastline. This one was caught off Wales, and when it came in I'm trying to get the date for you. In 2004, this was caught. That's not so long ago, but even then, molecular analysis was very difficult to obtain, and took a long time. And it took several months for this to be discovered to be a sturgeon species that had not been recorded from the UK. This was Acupensa oxyrhynchus, thought to be a Western Atlantic species, but here it was in the UK. And this gave a new perspective to initiatives to reintroduce sturgeon into the UK, because it seems from this specimen, it appeared we had 2 species present. Large specimen preservation at the limit of our capacity includes a giant squid. We have to protect ourselves from formalin, and so we have the right protective gear. On this occasion, there was a failure of the containment tank, and the formalin vapor in the room was very high levels, and it made life a little difficult. But we did preserve a whole Architeuthis 8-meter long giant squid. Next slide, please. Of the colossal squid, we only have Mesonicoteuthis. We only have the head and tentacles of this specimen, but here we are injecting it with formaldehyde. This was a northern bottlenose whale, which was stranded in the Thames in 2006. It caused a national media frenzy, and great crowds of people watched it because it took quite a long time to die in the river, and it was hoped that it would swim out to sea again. But when it eventually died, we needed to make a skeleton. We couldn't preserve the whole whale, and so all the curators at the museum converged on the specimen, and we spent a whole day defleshing it, and then the skeleton was sent for cleaning. So another important part of our job is hosting visitors from around the world. Here's Dr. Jayaram examining Francis Day fish skins in 1976 in our dried collection. I've just really selected a few almost random slides of visitors. We have so many, but it's great for us in our outreach. It's great for us to develop all these connections with the ichthyological community around the world, and it's really one of the perks of the job and one of the strengths of our position as an institution, that we can network with so many people from around the world. And this, of course, has been improved. Here's Dr. Bill Eschmeyer, the founder of the great catalogue, apart from anything else, the online catalogue, visiting in July 1981, great character. Gareth Nelson, such a monolithic figure in fish systematics in 1981. Here's Joe Nelson of the famous Fishes of the World, apart from all his other ichthyological studies, with Ralph in, what date is this? 2000, quite recently. And not, I'm afraid, sadly, but long before he died, 2006. Great to see him. Of course, no talk about ichthyologists can ignore Tyson Roberts, monolithic American figure who visited us in 2014. We are still in touch with him, although unfortunately he's in not good health at the moment. Here's Ollie Seehausen and Rosemary Lowe McConnell, one of the great ladies of ichthyological ecology, and this is working in our lab, the space I mentioned before. Next slide, please. Here's Lenin Chumbi from Peru in 2019. Next slide. Anouk with me in the collections in 2018. Victor Genevetti from Brazil. He's with Ralph here in front of our whole preserved marlin specimen, which I'll mention later. Next slide. Here's Unmesh with myself and Ralph, and he's standing next to one of our digitizing facilities. Digitizing being a word that's the trendy word for quite often photography, but the ability for us to supply quality photographs and radiographs has meant that we can put out collection information much more readily than in the past. Here's Chun Rashun Kamai working in 2018 in an evening presentation, explaining to the public about her work on Sicilians, and she really enjoyed the opportunity to meet a public audience in the UK. And here's Deepak talking to Simon Loader, the herpetologist in the museum. Okay, so looking at after our visitors is one thing, and of course, apart from the collections of specimens, the museum in London has a great resource in terms of literature and archives and records of all sorts. So this is a small library devoted just to fishes and herbs. And these are the books that are most used, most regularly, the standard and really in-demand works devoted to fish and herbs. There are other much larger libraries in the museum which contain runs of journals going 2 centuries back, and that resource is on paper, and it's a mistaken idea to think that it's all digitized. Our library is a resource which we have in the modern world to explain to people. We still need to keep those runs of journals. Next slide, please. Inquiries take a lot of our time from all walks of life, commercial inquiries, of course, largely research questions, but public inquiries as well. We get asked about the shagreen on a sword handle and show that it consists of prepared ray or shark skin, and that these denticles that are ground down in a prepared handle like this are dermal denticles that cover elasmobranch skins. These can help, if they're not overly prepared, to identify the animal from which the specimens or the samples of shagreen, the material that is used to make these ornamental coverings, we can identify the animal that was used to make them sometimes, and this is often required before export licenses can be obtained for antiques that are covered in shagreen, so that we can say that CITES protected species are not involved. Also, we are available if there's a good question to ask of our specimens to commercial inquiries. So the Speedo swimsuit company wanted to look at dermal denticles and think about and to take skin samples, in fact, and to test them for their hydrodynamic efficiency and try to copy the effect in ridges on a swimsuit. I mentioned radiography, producing X-ray images. We have a machine built in the 1930s, which we still use. We're encouraged to update this periodically, but we are rather obstinate in holding on to our X-ray equipment because it produces good images of a very wide range of specimen sizes, so that we can X-ray fishes of just a few centimeters long or a meter long. It's very flexible, and some of the digital systems that are now coming on stream, which will in the future no doubt be developed to the point where they can match what we can do now with this old equipment. We're not there yet. The new systems tend to have a very limited range of effectiveness over specimen sizes, and we do get good definition using film and this old equipment. We're not going to let it go until we have something that can cover the same ground. We do have a digital system, but it's not in fish. I have to say we still prefer the images that we get with this equipment. And here I am with a Noop, and we've developed in fluid baths this film, the fine grain film. Medical film won't do for us. We need this special industrial film with a very fine grain, slow exposure, and we get great results. So we can provide these images we can provide them digitally now. We used to have to roll up a sheet of film and send it in the post, but not only can we distribute these to our contacts around the world, but we're building up, of course, a bank of images associated with our database, so that it might be if you want to do vertebral and fin counts, you might find that the radiograph is already available in the collection. Another way of looking at the anatomy in a specimen, of course, is clearing and staining, and we have a large clear and stain collection. This is extremely useful in developmental studies, like the pufferfish that in the next slide, with which Ralph worked out the developmental processes in the development of the pufferfish jaws. And that method is really primary in discovering the anatomy of particularly small fishes. And we have a collection of those which can be referred to. We keep the material afterwards. So, incorporating new collections. This is the fruits of one of Ralph's expeditions with me to Myanmar. This is what we bring back in our suitcases, and this is the lots before they are sorted. Each jar represents a collecting episode or station, and then when it's sorted into the taxa, it expands. That lot expands into this, so you can see that as we get to the position where we can arrange the material systematically on the shelves, there's a great deal of expansion that takes place. We get increasingly, of course, requests for DNA samples, and I'm not advocating the use of an electric drill as a way of collecting DNA samples. This is a case of where it is practicable. My colleague James is taking a sample from rostrum of a sawfish, dried specimen, and drilling a small amount of tissue out produces a sample. Next slide, please. So where people asked for whole specimens, they sometimes now ask for tissue samples. We ask various conditions of the people who make these requests. We want to know your protocols, whether you basically know what you're doing, we don't supply the sort of volumes of tissue that were required in before 2000, as it's a good protocol now requires much less tissue. And we ask for aliquots to be kept and various other conditions. So you have to fill in a tissue sampling request form, but they are there. We like to provide tissue where it's possible. And we are also aware that people doing molecular studies have their own protocols. Those protocols are being refined all the time. So the notion that once a specimen is sampled for DNA, it never need be done again, we know that that's not the case. But we want to we balance those things, the rarity of the material, what you can do with it, the questions you're asking. And it's not our being intrusive or obstructive. We just want to make sure that the provision of tissue is sensible. Next slide, please. We have, of course, as workers with fish, contacts with several different aspects of society. So fish as pets in the aquarium industry, as food, of course, many inquiries from the food industry, but also sport fishing. So the British Record Fish Committee are a group that ratifies the angling records, as anglers are dead keen to catch the biggest, it's actually heaviest, that is the criterion, specimen of our perhaps rather depauperate post-glacial fauna in the UK. The hunt for the biggest carp goes on over the centuries, and every now and again a record is broken, and it's necessary to make sure that those records are accurate. And so when a claim is made for the record a new record specimen, and several claims are made every year, perhaps between 20 and 30 on an average year. The two of us in the committee, myself and the man to my left, are the scientific advisors who we will produce an ID, a reasoned ID that will say that the fish that's been caught, we say what species it is accurately. Next slide, please. And this is a Derbio, and we've the proud captor of this fish. It's quite rare, this species, Trachinotus in Trachinotus ovatus in the UK. Very few, just a handful of occurrence records. So this chap was able to claim the weight record for the species in the UK, and we got a fine specimen as a result, with good locality data. Next slide, please. So I produced the identification, and it's a reasoned identification. In the second section there, you see I put the diagnostic features that allowed me to come to that decision, and sometimes that's challenged, and it's always very interesting when that happens. But having the record of why the decision was made and the specimen itself for re-examination means that yeah, none of those challenges have been successful yet. Next. This, in fact, the captor is very proudly holding the first record of this species for the UK. So it's a record weight for because it's the first one, he automatically had the weight record. So he got a certificate, and nobody's caught that species since. So it's the record as an occurrence, and therefore obviously the record weight. Next species, next slide, please. Since my time in the museum, one has become increasingly and more involved with public outreach. We were relatively secluded in our basement in 1973. The new building that we're in since 2000, the Darwin Centre, was actually built specifically with exposure to a public audience in mind. And we run tours through the collections, guided tours, and here I am actually talking to the guides. So I tell the guides what's going on in the collections, and then they tell the public. Next slide, please. Here's an interesting specimen that provides a very valuable record, the second ever blue marlin to be stranded in the UK. And it also provided us with an exhibit in our newly opened, in 2016, we opened a new refurbished main hall, and we were able to preserve this specimen whole in glycerol. It's made a very fine attraction. You'll see visitors clustered around this tank on a day when visitors are allowed back in the museum. And this method of preserving in glycerol is relatively experimental. We took advice from the anatomical curator, Dries van Dam in the Netherlands, and we're monitoring it very closely. There were problems with mold growth initially, but it's very interesting that when a volatile liquid such as alcohol or potentially hazardous liquid like formalin are banned in public spaces, this presents a possible alternative for displaying whole fluid preserved specimens. Next slide, please. We have to help maintain the exhibitions, the specimens we have on exhibition. So our coelacanth started developing these lipid globules leaching out of the specimen, and visitors were saying they thought mushrooms were growing on our coelacanth. So next slide, please. We had to, out of hours, we had to brush these away and sort of just clean up the specimen to make it attractive to the public. And next slide, we achieved a cleanup. We'll probably have to do that in another 10 years. Next slide, please. We host a lot of VIP visitors. Here's the Queen talking to me about our swordfish specimen. The other side of that specimen is in a lot better condition. That's why we showed her that side. And this was on the occasion of the opening of the new building. Here's the Duchess of Cambridge, who's our patron, with Sir David Attenborough looking at the marlin on the day of the opening of the specimen, of the new exhibit. So thinking about the 48 years, the changes I've seen in the collection, one of the chief ones is the staff complement. So from 12 people in 1973, next slide, please. By 2013, by not replacing posts as they fell vacant, not recruiting, we were down to 3 staff. That's 2 curators, James McLean, myself, and 1 researcher in Ralph Bretz. And then in October 2019, we lost Ralph, our last fish researcher. And so there's just myself and James, the 2 curators. Curators is used as a slightly strange, in a slightly unique way in London. Curators are collection technicians, expert collection technicians. We have some involvement in research when we can, but we're basically facilitators for all investigations into the collection. And not having a dedicated fish researcher now is a severe blow to us. Next slide. We've also seen, I've seen the change of the museum's position in the government and in society. Initially, when I joined, we were very much a government organization, government funded. Here's our government logo. Next slide, please. And in 1981, it was our centenary of opening. We opened the museum in 1881 to the public. That's the new museum. Prior to that, we'd been part of the British Museum in Bloomsbury. The natural history collections were housed in this new museum and opened in 1881. In 1981, we were using this slogan, partly because of something that had happened 2 years before. Next slide. 2 years before, in 1979, along came Margaret Thatcher. And anybody who claims to fully understand British politics is either a liar or a politician. I'm not going to try to interpret exactly what happened, but it was clear that that Conservative government had a business drive and wanted to make business rules apply to previously protected government institutions, and that included we had to apply for more of our own funds, and we had to follow business rules. Next slide, please. Next slide, please. And that included rebranding. So here we've got a Tree of Life logo, which we actually call the zebra's ass, because once you've seen that as the backside of a zebra, you can't forget. It doesn't look so much like a Tree of Life after you've had that interpretation. And we started these rebranding exercises, which involved us changing all our stationery and corporate colors and following these strange codes about branding, which hadn't really featured in museum life until then. Next slide, please. And this rebranding exercise keeps going. Each iteration costs the museum more money, with business consultants firms designing new logos. Here's the N for Natural History Museum. Now we've changed our title from British Museum Natural History, and it's filled in with various pictures. Next slide, please. And then another branding exercise devises one without any content, and so on. This is just a new world that we live in. Next slide, please. The outreach is probably one of the good initiatives and good changes that we've seen come out of this new positioning. We have to encourage, we have to sell ourselves basically to a professional audience and to the public. And that has meant researchers describing their work to a public audience and getting more people in the country to understand what we do and why it's important. So this European initiative, European Researchers Night, the museum, because it loves branding things, had to put its own title, Science Uncovered, over the top of that. But yes, these were great events. Next slide, please. Very popular in the evenings where the museum was filled with a public audience. And I think it's fair to say that the people who came were people who were likely to be interested, and the sort of the feedback and the communication that we got out of this quite labor-intensive days were actually very worthwhile. Next slide, please. Yeah, here's Ralph and I presenting. The museum does love its mood lighting in the evenings, and that so you have to be careful to be able to illuminate your specimens in this atmospheric lighting. But yeah, a great deal of enthusiasm. I've also seen, of course, changes in buildings. So our beloved Waterhouse building opened in 1881, very sturdy structure as well as very beautiful. Next slide, please. The functional building of built in 1925, the Spirit Building, where the fish collection was housed, not designed to look pretty, very much a functional unit, but sturdy and built in an alignment and with no windows in one side to control environmental conditions passively. Next slide, please. And then these are one of the storerooms in, this is one of the storerooms in their old Spirit Building. Next slide, please. New buildings have a different take. They may have to attract business plans. So they are, sexy architecture is considered important, and functionality sometimes takes a bit of a back seat. Next slide, please. So the new buildings are built of glass and steel, and we don't know what their longevity is. Certainly large glass steel buildings built in the 80s, late 80s in London, some of those business headquarters, they are already starting to come down. So we don't have the information about the longevity of these buildings, but I suspect it's nothing like Waterhouse's building. Next slide, please. We talked about the manual work in the collections. Another change has been automation. So next slide, please. Spirit on tap now. No, we don't have to go and get it in jugs. So this potentially can speed up work, and there are efficiencies which I've definitely seen come in the maintenance of the collection. The trouble is, of course, that they require the technology to be robust, and I have to say the system breaks down quite often, and then we're back to the jugs. Next slide, please. The galleries that opened in the 1880s were a public attraction and an education. That's what Richard Owen intended. Next slide, please. And next slide, please. In the 1930s, J.R. Norman wrote a guide to the exhibition and refurbished it, and you see that it's specimen heavy. It's masses of specimens. You would get a very good grounding in ichthyology by spending time in this gallery and reading all the very informative labels and seeing how the groups were displayed on the walls all around a large gallery. Next slide, please. And J.R. Norman's guide to the fish gallery, which I purchased when I visited the museum as a kid, was originally published in 1937. 175 pages of quite compact ichthyological information. This is for the public visitor. This kind of depth and level of information is not available, it has to be said, from our modern exhibitions, which are intended, I guess, to reach a very broad audience. But yeah, there's been a loss of the opportunity to go into depth, I think. Next slide, please. Next slide. This is a modern exhibition opening this year. Fantastic beasts related to the children's author J.K. Rowling, involving mythical beasts. Next slide, please. So this is a specimen of supposedly of a horn of a mythical creature, and it's been made to look old, even though the museum is actually full of specimens that are really old. Weird and wonderful creatures exhibited, but alongside real specimens, there is an attempt to educate as well. But the sort of the central attraction is now mythical beasts, and this is a ticketed exhibition, so it's intended to make money. Next slide, please. Fantastic beasts again. Next slide. Okay, so the strategy for science has also changed. In 1981, it's very clearly set out here: 80% for science and 20% for exhibition and education. And it's a center for taxonomic research. So that's very clear. Next, and that's 1981, our centenary year. In 2013, we got a new science strategy. Next slide, please. And you can see the direction has changed. We're no longer primarily for taxonomic research. Next slide, please. Next slide, please. Now, our new strategy 2031 is a completely different thrust. Again, it's the planetary emergency. Next slide, please. And you can see the language has changed. We're not talking about systematics or taxonomy very much. Next slide, please. So yes, you can see that in the studies. This was produced by Peter Whitehead, and it's a 579-page monograph on the clupioid fishes of the world, which still stands as the reference work on the group. Next slide, please. In order to attract funds, you couldn't involve a monographic study like that. Now, these are the sort of studies that are being produced now. They're related to modeling, projection, and the Predicts project is talking about trends. And this one too, you can see that it's about modeling. It's about moving away from the data slightly and manipulating it. So thinking about what I've learned in my time at the museum, really the long view that collections should be and can be maintained to outlive trends there. And in order to do that, we've got to keep building them and maintaining them for not only the present, but for the future. And they will, as we see, we talked about all these changes that I've seen. There will be more, but the collections will still be there. So we could not have dreamt in the 19th century that we'd be looking for DNA in specimens, but now we can, and we wouldn't have had CT scanning as a way of looking at skeletons. And now we have, but only because the types and the original material are there. Next slide, please. So I think the fact that collections are bigger than individual researchers, even though their collections are built by individuals and mean nothing without individuals working on them, we have to have this longer view, and the people who fund us have to have this longer view as well. And when we pursue those funds, we shouldn't ignore that this is our central business. I think that's it. So thank you very much.